Thanks, Scott, and thanks, Tracy, for organizing this and inviting me to present with our model system. And my lab has been using organoids or iPS-derived neurotissue to really model neurological disorders, including epilepsy. And so it is a model of neurodevelopment. So we're really modeling the early stages of embryonic fetal development. And the basic idea was really to try to understand how the nervous system is formed. The fact that we don't have experimental ability to manipulate the human embryo makes everything complicated. And most of those conditions actually started in uterus. So how to reach the uterus. So the organoid to me is a model system that we can take the human brain outside the uterus in a controlled environment of the lab and recreate the early stages of neurodevelopment. So it doesn't mimic the adult brain, but it mimics how the brain is formed. And really the pioneer in this field was Yoshiki Sasai. So he first showed us that if you use human pluripotent stem cells and neuralize them in a floating solution, they will self-organize in 3 dimensions. And the cells will start to spontaneously differentiate and form tissues that are remarkably similar to the human fetal brain. Of course, it's a small scale, but nonetheless, I mean, these are some of the original pictures on his publication. It's quite remarkable that he was able to achieve that. So that publication happens exactly when I was starting my lab. So I thought, wow, that's something that we really have to develop. But unfortunately, all these protocols were quite complicated. What we see is that in the past, I would say 5-7 years is when most labs start to robustly adapt the Sasai protocol or creating new protocols. So it becomes like a more robust technology. But before I continue, I would like to remind you guys that what we call a brain organoid is definitely not a mini brain, a dish. So most people who are not familiar with the human system, they have this vision that we have a miniaturized brain inside the cells, and that's not the case. Moreover, there are several limitations of the model that before I continue, I would like to highlight. So first of all the protocols that we tested so far has very low neuronal activity, mostly because those neurons are quite immature, and also because we don't have an optimized condition to grow the cells. Everything that we had until recently was borrowed from mouse in biology, and human neurons and mouse neurons have different characteristics. For example, osmolarity of the cells is highly different. Still, some people use the same media for both mouse and human. So that tells you how the field is so young that there is so much room to improvement in here. These organoids are not vascularized, so that's why they don't grow too big. I mean, they are 0.5 centimeter in diameter. We are missing cell types. We don't have all the cell types represented. And depending on the protocol you use, you might have a little bit more diversity than others. As I mentioned, we don't know if we are growing them in the ideal culture condition. Reproducibility has been a nightmare for this field. I take the opinion that they are just recapitulating the heterogeneity of the human nature, but still there is experimental variability as well. And we don't know how translational they are. There's a couple of examples of findings in the organoids that could be translated in humans, but very few examples. Most of it, people were not able to validate or confirm. So these observations are still pending future validations. So we took the time to really optimize the culture conditions, and we create a new recipe to grow human cortical organoids. So we use the dual SMAD inhibitors as a transient exposure to the cells, not leaving them in the culture for long periods of time, but just transiently, so we can neuralize the cells. And then again, transient exposure of FGF and EGF, trying to mimic the human embryology to stimulate the cells to proliferate. And they will self-aggregate and form these spheres that you have here. And then finally, we just withdraw the growth factors without adding other factors and let the cells spontaneously mature, and they will round up, they will grow a little bit more, and they will start to differentiate in forming what we call the cortical organoids. If you just cross-section one of these organoids, what you have are ventricular zones similar to this one that starts as small neural rosettes. The progenitor cells are surrounding them. You can see the radioglia cells providing the cables for the progenitor cells to migrate and form the cortical plate. So as you age these cells, I mean, the number, as you age the organoid, the number of cells actually increase, and also the diversity of cells also increase. And this is a question that I get all the time. What kind of cells you have in your organoid? Well, it really depends on what stage are you looking at. We start from single cells. So the first cell is a human pluripotent stem cells, and we end up with 2.5 million neurons in there. From these 2.5 million neurons, if we just take a snapshot at 4 months, this is what we have. There's a pool of neuroprogenitor cells and intermediated progenitor cells that are differentiating to cortical neurons. Most of the neurons are glutamatergic neurons. At that stage, we start to see like a representation of upper and lower cortical neurons, and the diversity of cortical neurons will improve over time. The progenitor cells are also differentiating to glia cells, mostly astrocytes. We see that it's already present at 4 months. It starts when they are about 2 months of age, and then the glia cells will dramatically increase over time and will become 50% of the cells in the organoid. So it's 1-to-1 ratio between astrocyte and neuron. And that's the stage where we see a dramatically or exponential emergence of excitatory synapses in the organoids. So these astrocytes are not just there, they are actually promoting synaptogenesis. And finally, a very discrete population of GABAergic neurons. These cells actually, they have the markers for GABAergic neurons, but they're not yet GABAergic neurons. So this is also something that we see many people confound the presence of the receptors and when they actually become inhibitory neurons. So we classify the inhibitory neurons when they start releasing GABA, and we measure GABA in the media produced from these organoids, and they actually appear after 6 to 8 months of age. So there are a couple of receptors that are expressed, and they precede the actual function of these GABAergic neurons. So they would become functional just later on in the organoid. And that is actually very important down the road when we start to analyze the networks. So it makes sense actually when we see the evolution of these networks. And by the way, the networks is really something that we are very interested because the organoids as it is, it's a wonderful model if you are looking at transcriptional levels or the early formation of the neuroanatomy. But as I mentioned before, previous protocols didn't have that level of activity to really generate a network inside the organoid. So we were curious if we could actually see this kind of a network. And why this is important? Because if you can measure a network from the organoids, I think it's the first step to bridge from the basic fundamental biology that most of the stem cell people do to these neural oscillations where most of the cognitive neuroscience people have been correlating the different frequencies of these oscillations with cognition. And seizures is, to me, one of the most dramatic cases of that, where you can easily spot alterations in these neural oscillations that matches with the seizure of epilepsy activity. So that would be important for us to have the emergency of these networks, even if they are spontaneously in the culture. And there are reasons to believe that we could see, because we can patch neurons inside the organoids. And by the way, these are intact organoids. We were able to patch without destroying the organoid. So we can measure the hyperexcitability of neurons over time. This was great news. And we matched that with the gene expression. So all the ion receptors start to accumulate in the membranes, and that's why they become more active over time. And another good news is that the variability within the batches of organoids is not that great. So it's actually, to our surprise and kind of counterintuitive, is smaller than what we had in our traditional 2D culture system. So that was a good news. And but single neurons doesn't actually show what a network is all about. And this is just two examples. If you just take the analogy of an organoid with a stadium, if you're just looking for a single neuron or a single person here, you might not understand fully what's happening in the organoid. So we were looking for a method that we could analyze these networks longitudinally, and that's why we turned into the multi-electrode array. So this is a technique where we place the organoid on top of the electrodes. The electrodes are printed in the bottom of the dish, and we record the activity over time. So it's one of the few technologies that actually allow us to record for years. We have been doing that for several months and years now, and we generated an activity map such as this one, or a raster plot where you can follow the activity of each channel over time and see how it evolves. So that provides very rich data for us to start building network analysis. And so we were able to combine all the spikes into a spike raster plot, also creating like a population spiking that will give rise to what we call local field potential. And we use that local field potential as a proxy to see if these organoids will show any evidence of spontaneous neural oscillation. So long story short, we team up with Brad Wojtek, who is an expert on neural oscillations, and he got our data and was able to independently find evidence for these oscillations. And this is how they behave. By 2 months, we actually don't see neural oscillations. So if you just have organoids by 2 months, there is nothing there. By 4 months, that's when we start seeing a little bit of the formation of these oscillatory waves. And if you remember my previous slide, that's when the astrocytes start to kick in. So all the synaptogenesis actually happen when the organoids are about 4 months. By 6 months, we have the peak of synaptogenesis, and you see highly synchronized neural oscillations. But that will go away if you just wait a little bit longer and become highly complex starting at 8, 9 months. Again, that's when the GABAergic neurons start to become active. So it was very nice to see a correlation of the emergence of these neural oscillations with the different cell populations that we have in our organoid. And to our surprise, we could actually see that even in an organoid that contains 2.5 million neurons. So we didn't need like a full brain to have these oscillations. Just a piece of cortex can do it, which was surprising to me to see that. And how similar are these oscillations with the human brain? So since we cannot record from inside the uterus, we decided to take advantage of preterm neonatal babies. And these are babies that have EEGs. And we create a machine learning algorithm by training the machine with over 500 EEGs from prenatal babies between 25 and 38 weeks post-gestation. So once the machine was fully trained and can give the age of the subject by looking at 2 or 3 EEG features, we start feeding the machine with the data coming from the multi-electrode array. This is totally unbiased, so the machine doesn't know if it's analyzing the human brain or an organoid. And we ask, how good is the correlation? Can the machine guess the age of the organoid? This is one of the most surprising data that we had in the lab. The correlation is actually really good. I have the predicted age here with the actual age. So you see the human data in black and the organoid data here in blue. So there's a very nice correlation starting at 25 weeks that matches or are significantly identical to the human brain. Each dot here is an organoid. I'd like to call the attention that the variability in the organoid is higher than in the human brain. So it's not perfect, and we don't know where this variability is coming from, even though some of these organoids are genetically identical and will grow in the same way. But somehow the difference in these oscillatory waves becomes more and more diverse as we grow these organoids. We have some ideas of why this might be happening. All right, but what can we do with that? I mean, how can we use that model to study epilepsy? So we decided to turn into a genetic early onset epileptic syndrome. This is CDKL5 deficiency disorder to see if our model can recapitulate some of the features of that syndrome. And this is, it was published this year, so I'll skip most of the data. But basically what we find is that if you keep the organoids long enough, we see something that we never seen before in other syndromes, which is early stages of hyperexcitability followed by a plateau. And we can actually see that in single neurons as well. So this is recapitulated at the network level as well as in single neurons. And we first figured out, because we did the recordings at different stages, and we were not getting the same result. So we were very suspicious that, well, I mean, maybe we're changing the labels, there's something wrong here. So that's when we decided to do the longitudinal analysis and follow up with that. And that's when we learned that the syndrome do have this hyperexcitability when they are about like 25 weeks. And then it plateaus, whereas the normal brain organoid or controls continues to have that evolution of the networks over time, creating different oscillations down the road. So just breaking down in numbers, this is a number of spikes per minute. So the syndromic cells or organoid start becoming active very early on compared to the slow progress of the control neurons. So again, then we went back, we validated with single cells in old cultures, young cultures, and we were able to see that, again, this hyperexcitability, early hyperexcitability, it is indeed present in young, immature organoids. So what we think is going on is that the syndromic form of the organoid carrying the mutations in the CDKL5 gene actually produces a very early stages of waves that will take longer for a normal organoid to get to that stage. And once it gets there, it becomes overly synchronized and never advances to a more complex stage. So we don't think this is a seizure activity. We call that predisposition to a potential seizure activity. So we think that this behavior might, again, predispose to the epilepsy, as well as slow development that we see in this syndrome. The good news is that we can use this model to screen drugs, and this is what we have been doing. This is looking at calcium imaging or different characteristic of these networks over time. This is a spider plot. The black circle here is the control, and the red is the differences that we find for different characteristics of these neurons in the syndromic form. And here is when we try to fit the red into the black. So you can see that some of these drugs can actually fix some of these neuronal characteristics, but not all. So we never get like a perfect rescue, which to me is interesting because it suggests that maybe there is no single drug that can fix it all. Maybe we should aim for a cocktail of drugs, and that's another thing that we're looking for. Another good news is that since we know the gene, it's a single gene, you can aim for a gene therapy. And this is something that we have been playing with the organoids by engineer EEV viral vectors to carry the CDKL5 gene, and we infect the genes inside the organoids that are sitting in the multilateral array. And in this case, we are trying to put it down the activity by reducing excitability of these neurons. And with a single dose of AAV, we were able to do that. So we can fix that at very early stages in life, and the idea is to propose a gene therapy for young children with this syndrome. So another thing that I think is positive here is that our AAV is only targeting 10% of the neurons. So whatever is happening here is only happening 10% of the neurons. But thankfully, we are seeing like a dramatic decrease in the overall network, even though we are only correcting 10% of neurons. So, but what about the conditions that we don't have single genes? I mean, this is something that we are also trying to focus, and I'll give you an example. This is unpublished data for focal cortical dysplasia. We choose 4 patients. These patients are drug resistant, they try everything, nothing works. Surgery resistant, they passed 2 surgeries, they're still having lots of seizures. And the good news is that we're able to save tissue from the affected sites, as well as a little bit from the normal tissues. We did all genetic analysis, we find no obvious risk genes, and there is no mosaicism. We extensively look for mTOR mosaicism, and we could not find any mTOR mutation that will suggest a level of mosaicism. So we generate iPS cells from these patients, and it was a question mark. I mean, will they show any sign of focal dysplasia? Since we believe that this could be caused by an undetected mosaicism. And if so, maybe there is a germline alteration that we are not aware. So we generate organoids, we kept them for 150 days. During this time, we're collecting samples to do genetic analysis, as well as electrophysiology. And just some of the differences that we see between control and patients, the early alterations in the formation of these neurorosettes and organoids were quite obvious. We see breakdown of these early rosette membranes. And as the organoid becomes bigger, we start to see balloon cells, multinucleate cells, all the same characteristics that we saw in the tissue, suggesting that, yes, this is indeed something that's coming from the germline. We just don't know what it is. And it's not one of the characteristic genes. So, but for these 4 patients, we see something like that. And then we try to challenge them with an electrophysiological paradigm. Here, we are trying to stimulate and recording the organoids at the same time. So we engineer the organoids to carry channel rhodopsin, and we shine the blue light while we record from the organoids. And to our surprise, we could actually see here, a difference in response. While the control cells, they were able to buffer the stimulus very fast, we see this reverberation of some of these oscillations upon stimulus in the control cells, again, suggesting that there is some kind of alterations in those neurons that are not making them not really behave as they should. So this is about 2-month-old organoids. So these are mostly glutamatergic excitatory cortical neurons, and they are responding in different ways here. We are also taking advantage of that to really build the networks and trying to understand what's going on. So we create 2-dimensional models of the multi-electrode arrays that was captured in the organoid to create these network levels. So what we're trying to do now is screen for new drugs that can actually revert these network hubs and creating something that resemble more the control cells, so we could perhaps apply this drug to these patients. So I'll stop here, and these are my take-home message. I think that these organoids is a new model system that could be applied for the study of epilepsy, especially because they are mimicking both the molecular, cellular, anatomic, and now electrophysiological signatures of the neonatal brains. It's not an adult brain. It's a neurodevelopmental model, but it tells us how everything started. So the organoids mimic the hyperexcitability of a genetic epileptic condition, and we could use that to test therapeutics. And we also use for idiopathic focal cortical dysplasia, and we are hopeful that these new analysis will point to different medications for these patients. So these are funding agents. In just full disclosure, I'm a co-founder of Tismo, a company that uses brain organoids for autism and epilepsy. Thank you very much.